I am really happy to be here with everybody today and to have this chance to share our perspective on all the kind of great science and other advances that are happening now out in the Alzheimer's field that we think are really, really helping to improve treatment options that are available for people living with Alzheimer's disease. So Kelly already gave you a bit of my background, but I wanted to add a little bit more color to kind of what she told you about me. I am still relatively new to the Cure Alzheimer's team. I only joined back in May. It's been a really exciting and eventful and fast period of time in the last 6 months or so since I joined. And when I think back about my kind of education and my career path that you heard about over the past really 25 years, it looks a little bit twisty. And I want to kind of give you a common thread though, as I kind of take you through how I started and how I ended up where I am right now. There was really one common motivating factor for me behind each of my decision points, starting at the very beginning of my career. And that was my grandmother, who's shown here. This is Patricia Forg. She was shown here first on her wedding day, and then at a bit later in her life, which was more like how she looked when I knew her, of course, as my grandmother. She was diagnosed with Alzheimer's when I was in college at Michigan State University in around 1998. The treatment options at that time were almost nil. There was not much to be done, but Aricept had actually just been approved. And my mom and her siblings really had to work hard at that time actually to convince the neurologist to prescribe Aricept for my grandmother because it was so new. And her neurologist at the time was not even really aware of it or aware of a lot of the science behind it. So this was happening while I was in college. I had already been kind of waffling between being an English major. I wrote probably very terrible poetry, or majoring in biology, as I was also taking a really fascinating class on the brain at that time. That semester, or one of the semesters I was there not too long after my grandma's diagnosis, I remember writing both a poem about the amyloid plaques in my grandma's brain, as well as a research paper that summarized what we knew about how plaques formed in the brain. And it turns out it was very little, right? And that was really all it took for me. Scientists love unanswered questions. I actually love, Kelly, that you had on the slide the follow the science, because this is kind of one of the most important things, I think, in any scientific career is you find an unanswered question, you try to answer it, and then there's more questions. And so you keep kind of following that down the path until you learn something, hopefully. So I spent the next 20 years of my career on a pretty typical academic path, doing research, asking questions, getting funding on that, on that side of doing science, and publishing papers. I went through grad school at the University of Washington here in Seattle. I did a postdoctoral fellowship, as Kelly said, at the Gladstone Institute. And then I spent 10 years at the Allen Institute, really helping and learning a lot about groundbreaking kind of helping to advance and learn about groundbreaking techniques in mapping different cells within the brain. So I spent 20 years on that kind of typical path. About 4 years ago, I realized that after all that time doing science, I still knew almost nothing about how you actually take those data and those insights from academic research labs and turn them into drugs that are meaningful for patients. So I decided at that time to join a biotech company, Cahal Neuroscience, and to really try to learn about that process firsthand through living it rather than just reading about how you turn academic insights into drugs. And I learned a lot in my time there, but what I also learned was that drug developers kind of know what to do if you give them a good target. There's a pretty standard handbook if you want to have a good drug target for how to turn that into a drug. Doesn't mean that it's going to be a successful drug, but there's a pretty standard workbook there. But again, only if you have a good target. And we still really don't, beyond amyloid, which I'll talk about, we still need more and better targets in the Alzheimer's disease space to give to these drug developers to build new drugs. And those new and better targets come from new ideas and work by investigators at academic research labs. And that is really the reason that I came to the Cure Alzheimer's Fund, is that I wanted to kind of sit at this point where I could help to identify those investigators and those targets, and to help those people take their ideas and turn them into drugs eventually. So that's my color on top of my career path. So my plan for my talk today was to spend some time kind of on Alzheimer's disease 101 first, just to kind of bring us all up to the same level, hopefully, on some of the basics before I share some of the latest research that we are excited about. I guess what I'd start with here is that when pathologists look at the brains of people who had Alzheimer's disease after they've passed, they look for 3 hallmarks to confirm the diagnosis. The first hallmark is the presence of plaques made up of the amyloid beta peptide that form around neurons. There are also tangles of the protein tau that form inside of neurons. That's the black staining in the center image there. And then at the very late stages, there's also a lot of shrinkage of brain tissue that's caused by the degeneration or the death and removal of neurons from the brain. Now, these 3 pathologies together make up what we call the ATN framework that defines Alzheimer's in the brain. So the A for amyloid, T for tau, and N for neurodegeneration. There are also other common brain changes that are associated with Alzheimer's disease, notably inflammation in the brain that involves a type of a cell called a microglia. We also know that amyloid beta builds up in blood vessels and that there is a loss of connections between neurons at a structure called a synapse that allows for information to flow kind of correctly through the brain's networks. Now, those hallmarks are what pathologists can see in the brain after death, but really, we don't know, I guess, maybe the big question is what happens before then to cause a healthy brain to become that diseased-looking brain after death. We definitely don't know all the answers to this question yet, but we might be able to learn something about this process from knowing how clinical symptoms develop with age and time, and then try to relate them to those changes that are occurring in the brain. So Alzheimer's, from a clinical standpoint, can be a very long disease, and it's broken down into several stages that are shown here. There is first a sometimes very long phase of the disease called the preclinical stage, in which people really don't yet have obvious symptoms, but we know that there are changes that are already happening in their brains. When someone starts experiencing more obvious symptoms, they might be diagnosed with mild cognitive impairment. And if it's truly Alzheimer's disease, then they will become progressively worse in terms of their memory symptoms, as well as other behavioral symptoms that really begin to interfere with their own lives, their lives of their families, and the caregivers as well that are living with them or working with them. One of our clues to how a healthy brain ends up a diseased brain also comes from studies of the genetics behind Alzheimer's disease. From a genetic view, there are 2 forms of the disease. One are the cases of early onset Alzheimer's, also called familial Alzheimer's disease. These cases are much rarer. They represent only between 1% to 5% of all people living with Alzheimer's, and they result from inheriting a copy of a gene with a mutation in it that changes how the amyloid beta protein is made. Patients with early onset Alzheimer's start to develop symptoms before age 65, and in some cases where there's very aggressive pathology, this can even happen as young as 30. The second and more common form of Alzheimer's are the so-called late onset or sporadic cases. Now, in this form, genetics don't cause or definitively determine whether one will get the disease, but there are now about 30 different genes that are known to increase one's risk for developing it. And the most famous one of these that you may have heard of is called the ApoE4 gene. There are also a large number of these risk genes that are made by the brain's resident immune cells called microglia, which is why there's been a lot of recent interest, as I alluded to in the earlier slide, in how inflammation can contribute to Alzheimer's disease too. Overall, though, genetics has really shown us that dysfunction in amyloid beta related genes causes familial Alzheimer's, and that this leads to this kind of knowledge has led to the prevailing hypothesis that amyloid beta initiates a cascade of biological changes that cause Alzheimer's disease, and that this happens in both the early onset and the late onset forms of the disease. We also know now that genes are only part of the risk equation for late onset and sporadic Alzheimer's disease cases. Many other factors seem to contribute to the risk for getting Alzheimer's disease. So it's not just genes. The biggest one that I haven't even mentioned yet is actually age, but there are also a long list of other factors that impact risk that are shown here. And I wasn't planning to read them all out loud to you, but please feel free to ask me about any of these later if you have specific questions. I just want to highlight a few of them. So some of these risk factors, we should say, but not all of them could be changed or modified at either a personal or a societal level, perhaps, to reduce the risk of cognitive decline and dementia. And some studies have shown that addressing these risk factors when it's possible to do so may actually prevent or delay up to 40% of all dementia cases. That includes Alzheimer's disease cases. So the top 3 risk factors that have been identified in recent studies that are associated with development of dementia were midlife obesity, followed by physical inactivity, and then low educational attainment. Other groups of researchers have found that addressing some of these modifiable risk factors, specifically in midlife particularly, was associated with a decreased risk for dementia, even if you are an individual that has a higher genetic risk, like you might have that ApoE4 gene. And so I think the bottom line here is that while you can't change the genes that you've inherited, you may be able to influence the effect that they have on your brain by addressing factors that you can change. And in this case, earlier is probably better. So if earlier is better for prevention and even for treatment, as we're learning from some of the latest drug trials, we also need better ways to detect Alzheimer's at these early stages and to do this in a more accurate way. Here's where I think the field has made a lot of really great progress over the last 5 to 10 years. There's been a ton of effort poured into the development of tests that can detect changes in the brain using something called biomarkers. So biomarkers are really they're just changes that can be measured maybe more easily, that act as proxies for tracking the disease that's happening in the brain. The curves on this graph here show how different biomarkers like amyloid beta or tau, how these things change over time as they relate to the clinical phases of the disease. So one you'll see, just to point out here, is that the amyloid line, which is the red curve here, changes the earliest and it changes first. And so it's been a big focus of development of biomarkers. So people think that if you can detect amyloid at that early part of the phase, you can really potentially help to diagnose people that are going to, or predict who is going to develop Alzheimer's disease later. We do have multiple methods to measure amyloid in the brain. There's some methods directly imaging them with PET scans, and then also indirectly by measuring it in the cerebral spinal fluid, or even, I'll talk again about in a little bit, even in the blood. There's more recent tests where you can measure amyloid beta by taking blood samples. Biomarkers are really right now already being used to enroll people into clinical trials to find the right people that might benefit from the drugs that are being tested, and also to enable even earlier detection of Alzheimer's that would give people affected the opportunity maybe to address those modifiable risk factors when it's still early enough to do so. And then the biomarkers are also being used to help doctors diagnose Alzheimer's disease in the clinic. Oh. We'll skip over those animations. So those advances in the biomarkers have most definitely contributed to very welcome news in the field, which is the approval of the drug Leqembi just this past summer. Now, Leqembi is a monoclonal antibody treatment that targets and removes amyloid beta from the brain. You may know this already, but it's delivered via intravenous infusion every 2 weeks, and it's been approved to treat Alzheimer's disease in people with mild cognitive impairment or mild dementia, so earlier but symptomatic stages of Alzheimer's disease. People also have to, in order to be prescribed Leqembi, they have to have confirmation that there is elevated amyloid beta in the brain. So this could be done with that PET imaging or a CSF test. And Medicare in particular is requiring this in order for patients to potentially get reimbursed for the treatment. Now, there were serious side effects in a small percentage of patients called ARIA. This is evidence on MRI scans of brain swelling and some microbleeds. And we know that the chance of this side effect is higher for people that have that ApoE4 allele. So people that are taking or prescribed Leqembi will need to be monitored for ARIA with MRIs. And I end on this last point, which maybe should be the top point here, but just to say that Leqembi was approved because it did show that it lowered amyloid in the brain and slowed down cognitive decline in these patients by about 27% over an 18-month time period. Now, this reduction may be considered meaningful for people in the clinic or for people that are taking it and for their caregivers and their families, but it's clearly not enough. We're not done with Alzheimer's disease with this drug. Hopefully, I would also say that this slows the decline, but it doesn't stop it and it doesn't restore cognitive function. So we're not done with identifying new treatments for Alzheimer's. Hopefully, this is the first real crack in the floodgates of what will be, many new therapies coming down. So how do we get there to the new therapies? At the Cure Alzheimer's Fund, we think that, as we've talked about already, we think this will be through funding novel and innovative research at the earliest stages. And with the generous donations that we have received so far, we funded a really remarkable number of grants, I think, about 764. This was from the website, so the numbers look slightly different from yours, Kelly. I just took this right off there. But 764 grants to date, totaling over $182 million. So this has been, I think, incredibly impactful for the field. So our strategy in the research team is to really fund that great research, and what we fund and how we fund are kind of shown here. So I wanted to just touch on this again, although I think Kelly also covered this a bit. So what we fund is a diverse research portfolio that covers research into many of the complex mechanisms that are thought to contribute to Alzheimer's. We really look for great people and ideas at the earliest stages of research, doing foundational research, and these people come to us, and for the most part, through recommendations from our research leadership group, as well as others too. We fund research that allows investigators to leverage their work into larger grants and investments. We generally fund not-for-profit institutions around the world, but we can sometimes, in some cases, look into other funding of biotechs as well, for drug development or clinical trials. We also try to fund, though, research with that in mind that will benefit the entire field over just funding research for that enables the development of proprietary molecules or therapies. How we fund, you heard about our research leadership group, who provides us with the scientific expertise in identifying and evaluating the projects. And our awards, I thought we'd put up here, are typically around $200,000 per year for 2 years. And one of the main hallmarks of our approach is this high level of kind of personal engagement that we as staff internally have with our funded investigators, but also one of the things I think is really most impactful is actually having these active collaborations between our investigators as well. It really helps to seed, further ideas, new projects, and hopefully to really advance kind of our knowledge about the field and new areas that we think are most exciting. So currently we're funding projects in about 15 different areas, including some of the things that I touched on, like the genetic risk factors, the amyloid and amyloid beta, tau, ApoE, as well as the immune system and neuroinflammation in Alzheimer's disease. We also fund groups of investigators, our consortia, that are made up of about 5 to 6 different groups working together to attack some of the kind of most difficult questions and stickiest areas in the field. These include the ApoE consortium that's supported by the Fleming Foundation, a consortium that's also focused on brain entry and exit mechanisms and the role of tau on the neuroimmune and neuroinflammation response. And we have a brand new consortium that's focused on the role of the microbiome in Alzheimer's disease. So what do we think is next? Kind of get to the point. What's at the frontiers? So I want to highlight just 2 areas that I think are really active right now in the field and show a lot of promise in terms of the potential to reveal new biology and insights that could be druggable or for which we hope we can intervene. But both of these are still at a pretty early stage of research, so ripe for kind of funding that could accelerate their progress. Now, the first area is the interaction between the brain and the immune system. We used to think the brain was protected from the body's immune response. And while that's true to a certain extent, we're learning more and more about how these systems do interact and how it might go wrong in the case of brain diseases. So our funded investigators are asking questions like, when is microglial activation beneficial versus harmful? what is the role of peripheral immune cells in Alzheimer's, and why, one that I think is personally really interesting, why do vaccines just generally seem to reduce one's risk for developing dementia and Alzheimer's disease? Now, another exciting frontier that I mentioned on the last slide is at the intersection of the brain and the gut. So our stomach and intestines are colonized by millions of microbiota, bacteria, fungi, little critters like that. And evidence is growing for a role of this microbiome in mediating brain health. But so far, it's been really fuzzy exactly how these systems might talk to each other. So our researchers in this new consortium are asking questions about how gut inflammation and the specific kind of makeup of the microbes that have colonized the gut, how that affects Alzheimer's pathologies like amyloid and tau, and also looking into how whether other cell types like the microglia might be involved in mediating this communication. So this is, I think, one of the most fun stages of doing science, exploring these kind of new ideas and new questions. And we can never, of course, as scientists and as a funder, never promise that it's going to lead to a new drug, but we have a lot of confidence in these investigators that we have funded here. They're all excellent, rigorous, and productive scientists, and all of the science, again, was reviewed by other scientists in the field to kind of ensure rigor and careful design of experiments. So I think they're as good as anyone's going to be at kind of attacking some of these questions. So I have 2 more slides to end my overview before we can hopefully have a fun and more interactive discussion, because I do find myself, I think, in a surprisingly optimistic frame of mind, maybe for the first time in 15 years, about the state of Alzheimer's treatments. I would say that for maybe a step back for a second, say for many years, my family always turned to me and asked me, you know, what's new? When is there a drug coming? And I felt like I was always a downer at those, like, you know, the family gatherings. Like, there's nothing. There was another clinical trial failure. There's no way to detect it. Now I don't feel that way. I feel really excited to go out and talk about all of the new advances. And I think we're finally in a really good and exciting place for Alzheimer's treatments, because we've seen big advances in different tools. So one of the advances are in the better diagnostic tools. We have blood-based tests for amyloid beta that are coming online now, which are going to be more easily accessible to patients than a PET scan or a spinal tap. These are available for clinical use already, and more and better versions are expected soon. I didn't talk much before about biomarkers other than amyloid and tau, but there are also recent advances in developing ones to monitor for inflammation as well as synapse loss. And we also have now 3 different anti-amyloid immunotherapies that have shown a benefit to patients in phase 3 clinical trials. Leqembi, that I highlighted before, is the only one right now with full approval, and donanemab, which is a similar drug from Eli Lilly, is under review by the FDA now, with a decision expected, and they just pushed this back to Q1 of 2024. The other thing I wanted to point out was, although these are all infusion therapies right now, there's also a lot of effort to see if subcutaneous injections of these antibodies might work as well as the IV infusions. And then after those 3, there are, you know, many more drugs that are working their way through clinical trials. This bullseye picture shows all of the drugs targeting Alzheimer's disease that are in that are in clinical trials. So the outer ring are drugs that are in phase 1 trials. The middle ring is those that are in phase 2, and the center are drugs that are in phase 3 clinical trials right now. There are 151 active trials ongoing. So that's, I think, actually great news, with 120 drugs being tested. This also means that there are a lot of participants needed to make sure that these clinical trials can be a success. So some of these have already enrolled patients, and some have yet to enroll patients. There are both prevention and treatment trials in this bullseye here. And the other thing I want to point out is that the targets are diverse. They're not just amyloid related, which would be the red ones here. There are many other targets that are making their way through clinical trials as well. So I think this is a great reason for optimism as well, that some of these are going to make it through and maybe be better than Leqembi. The last thing is that you don't have to wait for drugs either. As we talked about earlier, there are some things that you can do now. This acronym SHIELD is courtesy of Dr. Rudy Tanzi, who's the chair of our research leadership group. his recommendations for kind of prevention and protecting yourself include sleep, handling stress, interacting with the world, especially other people, so maintaining those social connections, exercise, learning new things, and to keep an eye on your diet, I guess, for more fiber, less red meat and processed food, less of what we consider this Western diet. The other thing we want to say is right now, get your vaccines, not just COVID. This is very early work, but so far the work seems to suggest that vaccine uptake in general, also for some still unknown reasons, seems to reduce risk for Alzheimer's and other dementias. So thank you for listening.